You're probably familiar with a social network. It's the web of connections between you and the people you know. Marketers have mastered the art of decoding these connections to target advertisements specially tailored to their audiences. Now scientists are asking if we can analyze a cell's genetic networks in the same way to find new drug targets for cancer and other diseases. A new computer program tracks conversations between genes in the same way that marketers track social networks. The genes in our cells talk to each other, and like members of a social network, some talk to each other more than others. Some a lot more. When genes talk to each other a lot, they form natural groups we call communities. In a healthy cell, gene communities function like an efficient factory, taking in raw materials, processing them, and manufacturing goods the cell needs to thrive. In cancer cells, the conversations can get weird, like if your boss started calling your best friend's boyfriend to cover a shift. These weird changes could be pretty awkward in real life. In cells, this leads to defective products and disease. You probably don't have 20,000 family members, coworkers, and friends. But cells, with 20,000 genes apiece, have a way bigger social network than you do. So when you map out a cell's social network, you get something that looks like a hairball, compared to the relatively tidy map you can make of your own network. Understanding how conversations between genes change as cells shift from healthy to diseased states can provide clues about how cancer forms. But with so many genes in our chromosomes, detangling the hairball of connections is a massive undertaking. To make the process easier, researchers from the University of Arizona Cancer Center and Dana-Farber Cancer Institute have created a new approach that uses the tools of network analysis to investigate disease. It's called Altered Partitions Across Community Architectures, or ALPACA for short. Yes, as in the cute furry animal. ALPACA is different from methods used in the past that looked at just a small portion of the hairball at a time. Instead, this method looks at the whole picture, focusing on finding conversations that happen in diseased cells, but not in normal cells. Identifying gene communities that are active in diseased cells can guide researchers in developing drugs that target those communities. The full potential of network analysis is only just starting to be understood, but the field is growing quickly. Scientists at the University of Arizona Cancer Center are developing new ways to analyze how networks drive disease.